Hello, I'm David Kerr, Professor of Cancer Medicine at the University of Oxford. I've just come back from lecturing in China with some fantastic collaborators of ours at Huaxi Hospital of Sichuan University. One of my lectures was innovative trial design, thinking a little about how we bring innovative medicines into the clinic from the laboratory base to the patient's bedside. And what I'd like to do is to bring to your attention a fantastic review published fairly recently in Annals of Oncology, looking at some of the recommendations from this task force that was set up by FDA under their auspices to look at the methodology for the development of innovative cancer therapies, MDICT. I've been a cancer doctor for 40 years. Had though that may be to believe looking at my relatively youthful potato head. And in that time, I have seen some very interesting ideas as to how we take new agents forward into phase 1 trials. In terms of targeted therapies, they're not quite fit for purpose. Principally, the classical aim is to define the maximum tolerated dose, giving us a feel for what the pattern of side effects and toxicity of the drug is, perhaps link that to pharmacokinetic endpoints or increasingly pharmacodynamic endpoints in which we're using biopsies or taking normal tissue surrogates to see if we can demonstrate that the drug hits its target, that sort of thing. With targeted therapies, we quite often get toxicities wrong, and in terms of defining a biologically optimal dose, the drug of dose which in the target tissue, tumour in our case, inhibits the target as effectively as is necessary to change the phenotype of the cancer cell, usually induce cancer cell death. So we've seen innovations in which we've looked at pharmacokinetic dose escalations. These sort of failed, generally speaking, because when we look at the pharmacokinetics of a drug, in cancer patients, certainly the inter-individual variation, the difference between dosing between you and I is very large, and it makes it really hard to come up with a target pharmacokinetic parameter, steady state concentration, area under the plasma concentration time curve, that allows us to dose escalate rationally and safely. I'm a bit of a pharmacokinetic nerd, which is why I really would have loved that to have worked. There are other schedules like intra-patient dose escalation, which is sort of good. We've looked at truncated methods. We've looked at different statistical methods. The statistical underpinning of phase 1 trials has always been threadbare, to say the very least. But we've used various other statistical models using prior assessments to try and speed things up. They weren't very impactful, one would have to say. But worthwhile, for those of you that are interested in new anti-cancer drug development, which should be the whole oncology community, worthwhile having a look at I actually find rather thoughtful recommendations coming from this review. I guess the bottom idea really was that rather than being able to define naively a single dose to go forward to phase 2 trials, perhaps we should try to recommend a range of doses which we might be able to test in a rather more adaptive or even randomized phase 2 trial design. I've said this many times before, the single most important thing that as oncologists we can control about the treatment that we deliver to our patients is not their germline genetics, it's not the somatic tumoral mutational landscape, it's the drug dose and schedule that we give. We can control this, but we need to put more thought into it. We need to think more strongly about the statistical underpinning of phase 1 trials. And I agree with the authors of this report, with the general thrust of the task force, that looking at a range of different doses will give us better options about defining, particularly with targeted therapies, a more rational dose schedule to go forward into phase 2 or emerge from phase 2 into phase 3. Have a look at the review. Tell me what your own thoughts are. For those of you who are interested in the tricky methodology of phase 1 trials. I'd be very interested in your comments. But for the wider oncology community, you must see how important it is. And I'd like all of you to think about those and think every time we treat a patient, whether with drugs, relatively rarely in single dose or in combination, are we certain that we've got the optimal drug dose schedule? Have a think about it. Any comments would be most welcome. But for the time being, Escapers, as always, thanks for listening. Over and out. Thank you.